Thank you very much, Bengt, for the kind introduction. Also, thanks to Shu Wang for inviting me here. It's so great to be here at these Molecular Frontiers events. They are fantastic events. So wonderful to see also so many young people. And so maybe before I start my talk, just a little bit of reflection back on my own career. I'm a chemical engineer of training, like several others that we've heard from before. When I took my engineering degree, I was kind of certain that I was going to go into industry and work. I come from a family of a non-academic background. I was actually the first one in the family to go to high school. So I was certain I was going to go into business. But I had a very persuasive thesis advisor that convinced me that it would, academic track could be interesting for me to pursue, and so I did pursue that and was in academia for a long time. However, throughout my career, I've always been interested to see how we can translate the research that we are doing into some use for people and society, and how we can also help maybe existing processes, existing technologies, and so on to advance. So this has really been a key driving factor for me throughout my career, and I'll also address some of these points when we move on. The title in the book says Metabolic Engineering. That is basically how to study and engineer metabolism, and I'll tell you more about how we can get to that. But first, a little introduction to metabolism. We heard from the previous speaker how complex a cell is. That definitely also goes for all the chemical reactions that are occurring inside a cell. And metabolism is really the core of all cellular function, because the metabolism is ensuring provision of the free energy to ensure that all processes can operate. So that's in the form of chemical energy. But also it provides the molecular building block that is necessary for the cell to synthesize its different components, its nucleotides for DNA, RNA, its amino acids needed for protein synthesis, and so on and so on. So it's really one very large network, as I'll show you later, that is behind, you can say, the core of a cellular function. But let me start a little bit with an introduction and say, how did we acquire all this information? And it actually goes back to about 100 years ago, when people were beginning to study the fundamental pathway that we normally refer to glycolysis, namely the conversion of glucose into pyruvate, and in human cells onwards to lactic acid. And so in 1922, Otto Meyerhoff obtained the Nobel Prize for his discovery of some of the individual steps in that pathway. And as we know today, there are many different chemical reactions that are occurring in this pathway. Each of these steps is catalyzed by an enzyme that is, of course, encoded by a gene. And actually, for many of these steps, there are several enzymes that can be isoenzymes, as we call them, that actually catalyze the same reaction. Emden and Parnas were also key contributors to this discovery, so we generally refer this to today as the Meyerhoff-Parnas pathway. Later on, Otto Warburg discovered oxidative metabolism, so that's the oxidation of pyruvate all the way to CO2. In connection with that, electrons are transferred to an intermediate compound we refer to as NADH. Shortly after, or some years after, Hans Krebs discovered the so-called Krebs cycle, or the TCA cycle, for tricarboxylic acid cycle, that is in describing all the individual steps that takes pyruvate and oxidize it completely to CO2, and it's actually a cyclic process that regenerates some of the compounds that are used in that pathway. The cell, of course, needs to regenerate this NADH that it generates, and that was described by Peter Mitchell in his so-called chemiosmotic theory, that describe how that is oxidized, and in the same process, protons are pumped across a membrane, and that creates an electrochemical gradient. And later on, it was also shown that when these protons re-enter, in this case crosses that membrane barrier again, it goes through an ATP synthase that then generates ATP. And that was already proposed in Peter Mitchell's chemiosmotic theory, but it was Boyer and Walker that got the Nobel Prize in 1997 that actually discovered and clarified the structure of this extremely complex enzyme, ATP synthase. ATP synthase is a multifunctional enzyme, and it's at the size almost of ribosomes, so it's extremely large, and it's embedded in membranes, and it's carrying out this very, very important function. So today we have mapped, practically all the different chemical reactions that are occurring inside the cells. We know also in most cases which enzymes are associated with that. Or at least that's what we believe, and I'll come back at the end that there is an aspect that we have often overlooked, namely the promiscuity of enzymes. That was mentioned also in the previous talk about the promiscuity of proteins, and that actually opens up for even wider metabolism that we have seen before. But overall, we actually do know this. So maybe also another brief introduction about metabolism is that if we look at yeast metabolism, which is something we have been exploiting for millions of years, or no, thousands of years at least, in human civilization, where glucose is converted into ethanol. And that's happening when there's no available oxygen. And in that process, of course, we get this conversion to ethanol, which is used in alcoholic beverages, but it's actually also what is used in leavening of dough, because that gives very rapid production of CO2 that gives the leaving of the dough. So this yeast can do very, very efficiently. Yeast also can oxidize pyruvate completely to CO2. It does that in the presence of oxygen. But I'll come back to later that there is actually a twist, because when yeast grows very, very fast, even in the presence of oxygen, it also converts glucose into ethanol. If we look at the similarity with human cells, for example, a muscle cell, it's very similar. We have glucose, but glucose is not converted into ethanol, it's converted into lactic acid. And we are very well aware of the phenomenon that happens when we don't get enough oxygen to the muscles. You produce lactic acid, and that starts to give sore muscles, of course. But the muscle cells can normally, if they get enough oxygen, get even more energy out of that glucose and oxidize it completely to CO2. Interesting enough, the conversion of glucose into lactic acid also goes to lactic acid, even in the presence of oxygen at certain conditions, particularly when the cells are growing very fast. And that's referred to as the Warburg effect, and I'll come back and talk a little bit about that also later. So some really big similarities, you could say, in terms of structure and also evolution of two different pathways. So if you look a little bit about yeast, that's been used for millennia as what we today refer to as cell factories. We use them to produce bread, beer, and wine. Later on, from 1915, during World War I, it was used to produce glycerol for explosives. Later on, we started to use yeast to produce ethanol in very large volumes, and more later you see application and using yeast widely for production of pharmaceuticals that are used both as vaccines but also recombinant proteins like, for example, insulin and others. And today yeast is also being harnessed for production of many different chemicals that we originally derived from nature, for example, from plants. It can be artemisinic acid. It's an anti-malarial drug. It can be farnesene, that is a nice-smelling molecule that is used in many perfumes and fine fragrances. We really are using yeast extensively, and it's also been studied extensively over the years. But it was actually, and even though it was used for thousands of years for production of bread, beer, and wine, it was actually first Louis Pasteur in 1876 that discovered that yeast is the underlying cause for production or conversion of sugar to alcohol. And so today, as I mentioned, yeast is really used widely as what we call a cell factory, particularly large volume production of ethanol, but many other compounds are also being produced, and particularly of interest may be today many of the advanced pharmaceuticals, like, for example, semaglutide, that is an ingredient in Novo Nordisk new wonder drugs for obesity and insulin and diabetics, is where the active ingredient is produced also using a recombinant yeast. And we see that in many other applications. There are also many other applications of yeast. It's also, I just saw recently that the first scale production, large scale production for producing of opioid substitutes, for example, for like morphines and others, could also be produced in recombinant yeast. So that's what we've been working on in my lab for more than 20 years, where we engineered yeast to produce many different chemicals. We looked into, you could say, the fundamental hypothesis: can we modulate metabolism such that we can produce chemicals of interest to us in our daily lives? That goes from everything as you could say pharmaceuticals at the bottom, all the way up to commodity ethanol used as a biofuel, but also many other compounds that can find specific applications as either solvents or also as what we call fine chemicals that are used, for example, as ingredient in food, feed, or, for example, also perfumes and fine fragrances. We also worked extensively on developing the methods to do this. So for engineering the cells, that's often referred to as synthetic biology today, but also developing systems biology tools that can allow us to find out how can we engineer these organisms efficiently. So one example I just want to mention is we worked for many years together with the Swiss company Firmenich. They have now merged with a large Dutch chemical company, DSM, so DSM Firmenich, about engineering yeast to produce a class of molecule referred to as sesquiterpenes. This class of compounds is used widely in perfumes, and they were interested in having a yeast-based production of these, even though you can extract them from plants. And the reason for that is that in some cases it's difficult to get enough material from extraction of these plants. Some of these plants are extremely rare, hard to grow. You cannot grow them in greenhouses and so on. And so therefore, if you have it in a yeast-based process, you can have easily scale, and you can basically produce on demand the amount of molecule you want to produce. We worked particularly on engineering yeast for production of xanthine, that is shown there to your right, and that has now been scaled up by Firmenich and is a commercial product today. And so in many of the perfumes you'll be buying today, they are actually produced using recombinant yeast, some of the work that we laid the foundation to. Besides the interesting application of yeast, it's also interesting to note that it has really served as a very important model organism. So what is shown here is a number of pathways that play a very important role in terms of overall regulation of cell growth and proliferation. And so you see here we have pathways involved in energy generation like glycolysis, beta oxidation, mitochondrial biogenesis. We have at the center processes like translation, lipid biosynthesis, amino acid biosynthesis that are really revolved in you could say, overall cell synthesis. And we have over here processes that are related to cell death, degradation of proteins and so on. And these are tightly regulated, these different processes in the cell at any time, and they are particularly regulated through integration of two key proteins, AMPK and TOR, and these are sensing nitrogen and glucose, basically, or energy. And so hereby, depending on what is the nutritional, the amount of nutrition available to the cell, these are regulated in order to say, okay, we have a lot of nitrogen, for example, let's begin to proliferate and grow fast. And that's also why, for example, if you don't have nitrogen, you will start to onset autophagy, for example, and you will start to degrade proteins in the cell and stimulate specific processes. What is also shown here upstream of AMPK and TOR is other regulators that are known, and these are playing a very important role in regulation of development of cancer. And so obviously, as soon as we get an imbalance in regulation of these two key components, the cell kind of gets out of control and starts to proliferate fast, and you develop cancer. What is interesting to note is that many of these processes here were originally identified in yeast and were later found to be completely homologous in human, and is now playing a very important role in our understanding of our cancer biology. So therefore also, people are talking about noble yeast. There's a number of Nobel Prizes that's been given out to fundamental biological processes where people actually studied yeast. We have Tim Hunt here. He keeps telling me that he didn't work on yeast, but his two colleagues that get the Nobel Prize did work on yeast, namely Paul Nurse and Hartwell. But also many other processes were discovered. For example, a very important pathway for protein secretion that is also involved in a number of different disease developments was also discovered during yeast. And more recently, in 2016, Osumi got the Nobel Prize in medicine for discovering the mechanisms of autophagy, and he had done all the fundamental work in Saccharomyces cerevisiae. The other listed below are where yeast to some extent was used also in their work in discovering some of the key processes. So studying yeast is obviously, as I said, a good model organism for understanding a complex cell, but it has this, can say dual purpose that we can also use yeast for engineering to produce interesting molecules. And so therefore, we have been looking very much on, could we begin to develop tools for moving beyond on studying individual pathways, but actually the whole metabolic network of a cell, and classify and incorporate that into a framework that allows us to discuss or to evaluate how the many different processes interact. And so this is where we have been working on using mathematical modeling. And there you can of course see the clear analogy and my engineering background, the objective of getting to a state where we can begin to use computer-based design of new cell factories and hereby speed up the development of new processes going forward. I completely agree with what has been discussed earlier also. We will probably never be able to comprehend and grasp all the fundamental processes and describe that in mathematical formulations, because it's simply too complex and there's too many processes that we don't know of. But from an engineering perspective, that also doesn't really matter. Most models used by engineers are simplifications and applied for that purpose that you want to apply it for. The modeling framework we're using, we refer to as genome-scale metabolic models. What we do is that we compile information about all the metabolic reactions that are occurring inside the cell. So we write down the stoichiometry of these reactions here. A goes to B, it's using a cofactor ATP here. It's catalyzed, actually this reaction here is catalyzed by two different enzymes that do the same reaction, so to speak. And they are of course linked to a gene. Further on, we see here another chemical conversion. B goes to C, C goes to D. In this last step here, it's actually a protein complex that carries out that reactions. There's a very important distinction between this function and this function up here, because you can lose E1 and this pathway can still function because E2 can replace it. Whereas if you lose P4, this pathway cannot function anymore because E4 is not functional. It's only functional when both P4 and P5 is there. So this distinction between protein complexes and isoenzymes that have redundancy, so to speak, in their catalytic capabilities is quite important, and that we are mapping out very clearly. When we do this globally, we'll get a large metabolic network. I'll show you one, but we also have a mathematical representation of the stoichiometry of all these reactions, and that's what we can use for mathematical modeling. So one important revelation that this brings us is that metabolism is not organized in pathways. This is a connectivity graph where all the individual metabolites that are catalyzed or used by different enzymes are shown as the colored dots, and all the green dots here are the enzymes that are addressing and incorporating and working together with these metabolites. And if you take, for example, a molecule like pyruvate I talked about earlier on, that's used by something in the range of 20 or 25 different enzymes in the cell. You can take another one, glutamate, an important amino acid. It's used by more than 50 enzymes in the cell. And so obviously there's this strong competition between the different reactions, and their function needs to be tightly coordinated in order to get this to work. And whenever I look at this, I always get amazed, actually, that it works all the time in our cells to a perfection, you could say. And that's, of course, because it evolved to have this. And this regulations I talked about before, AMPK and TOR regulation, is, of course, on top of this and making sure which pathways should be turned on and turned off in response to changes in the environment. So with the modeling, we could begin to model this, and I'll come and give some examples of this. But there is one thing that we did not include initially, but we have started to include, and that's information about the enzyme kinetics. I've always been reluctant about adding too much kinetic information because we saw a figure in the previous talk about how it is inside the cell. It's extremely compact. It's not like when we are studying an enzyme in solution and we have the metabolite freely available around that enzyme. It's an extremely compact, maybe it doesn't even make sense to talk about the concentration of free-flowing metabolite inside the cell. But there is, of course, one important constraint, and that is that all the enzymes don't come for free. There is an upper limit to how much conversion as a given enzyme can carry out, and that's determined by two factors: the amount of enzyme inside the cell, and the second, the catalytic efficiency of this enzyme here. So this is the kcat, and here's the enzyme concentration. So the flux, the reaction rate of going through here has to be constrained upperly by this value here. We don't know if we have these determined kcat from experiments, often done in solution with isolated enzymes correctly, but at least this provides an upper bound for the function of these enzymes, and this becomes quite important because it actually helps us to constrain these reactions here. So before I get to some illustration of how we can use these models, let me just also talk a little bit about how comprehensive they are. So if we look at yeast, and this model is referred to as yeast 8 because it's the 8th generation of models that we have developed. And so what we have here in yeast 8 is it's comprising almost 4,000 reactions, chemical reactions inside the cell. These reactions are associated with about 1,100 enzymes that are encoded by specific genes, and we have about 2,700 metabolites that are engaging in these different reactions. So you can see this is way beyond what we can comprehend sitting on a paper and a pen. This has to be done using computer simulations. Maybe one important note also that what we have done with this is that we have taken lessons from software development in terms of continuously updating. So whenever one of my students or anyone in the yeast community discovers that we made a mistake, because of course we did make some mistakes when we comprehended 4,000 chemical reactions into a model, there was maybe a mistake. It can be reported and it can be tracked who reported this mistake, and that can then be connotated into a next version of the model, and so it continuously updates. So it also becomes you could say, a platform as a library for information gathering throughout time. So let's get back to the Crabtree or the Warburg effect. Actually, it was studied also by Crabtree. A little bit interesting. It was actually Crabtree that discovered that cancer cells produce lactic acid. But he later got more associated with the process of why yeast produces ethanol even in the presence of oxygen. And you could say, why would a cell want to do that? Particularly, and as I mentioned, it happens when the cells are growing fast, because you can see, if you look at the respiration, you get a lot more ATP per glucose molecule, and you kind of see ATP as a key energy factor for driving growth. We get much more ATP during respiration or oxidation than we do when it's converting glucose into ethanol, for example, or lactic acid in human cells. But there is, you can also clearly see here, it's much simpler pathways. I mean, up from glucose to pyruvate, it's the same for both. But here, if you want to go to ethanol, you just need to use 2 enzymes. And actually, if you need to go to lactic acid, you just need 1. Whereas if you need to respire pyruvate or oxidize it completely, you need all the enzymes of the Krebs cycle, you need the whole respiratory machinery, and not the least, you need this mega large enzyme ATP synthase. And so these proteins, this is a lot of protein that you need to synthesize in order to capture this efficient energy here. And you can see already here, there are some indications that it is more expensive, of course, to get to the high efficient pathway. So when we looked at this from a modeling perspective, and we did this actually just to illustrate the point, both for yeast, Saccharomyces cerevisiae, and for a gut bacterium, E. coli, we gathered information about the amount of ATP produced if it's so-called the high-yielding pathway, namely the respiratory pathway where you oxidize glucose completely to CO2, and what we call the low-yielding pathway, that's the fermentative pathway. And here we calculated, using the model, the exact amount of ATP generated in 2 pathways. We collected information about proteome from measurement of all the proteins inside the cell, listed here, and how much is engaged in each of the 2 pathways. And then you can see here that the protein efficiency is different. So the protein the efficiency, you get more ATP per unit of protein mass in the low-yielding pathway, both for E. coli and for yeast. It's not a lot, but it is something. And so here's what is interesting, is that when the cell grows faster and faster, what it particularly needs to grow faster is ribosomes. Ribosomes are required for translation of the messenger RNA into proteins. The ribosomes are quite large, and it's quite energy expenditure also. But what happens is that the cell needs to compromise about where it should produce its protein. So more ribosomes, or should I invest into energy metabolism? And here the cell found a way that it can actually make energy efficiently by respiring or fermenting, just going to ethanol, or in E. coli's case to acetate, or in human cells to lactic acid. So you get more ATP per amount of protein. So it's really the efficiency of the protein mass that matters. And so this completely explains the Crabtree effect and the Warburg effect, something that people have been puzzling about for a very long time. What is regulating this, and why are the cells doing that? It comes down to a very, very simple explanation about protein efficiency. You see this very clearly here also. If you map here the amount of protein allocated, interesting enough, you can see here the amount of protein allocated for energy metabolism is surprisingly constant for really varying uptake rates of glucose. So no matter how much cells are taking up glucose, the protein allocated for energy metabolism is about the same. But what you can see here is that here you see that it plateaus here, and that's this when in the beginning here, you can use respiration because you don't need to turn into this, but because you have this upper bound on protein allocation for energy metabolism, when you come up in high glucose uptake rate, you'll just turn it into ethanol, or in E. coli's case, acetate. So with this model here, and I'm not going to go into great details, but we can actually describe many different phenomena. We can describe how yeast is growing on many different carbon sources. And this is a little bit interesting, because from a stoichiometric point of view, there's no difference between galactose and glucose in terms of the amount of ATP you get out. So why is yeast growing so much slower on galactose? Well, the model can actually predict this very, very precisely because the catalytic efficiency of the galactose consumption is lower than on glucose. And as the catalytic efficiency is lower, therefore you need more protein for that. That has to be stolen from somewhere, so to speak, and that's from the ribosomes, and therefore yeast can only grow slower on galactose. And similar for other carbon sources, and the model predicts this just as matching experimental data very, very well. Interesting also, if you do an experiment where you take express an inert protein inside the cell, and in this case here, it's just a green fluorescent protein or yellow fluorescent protein. It doesn't do anything. It's just being produced. But the more you produce of it inside the cell, the slower the cells are growing. And the model can also capture this. So this is just another way you could say of stealing protein mass for an inert, in this case an inert activity, namely a yellow fluorescent protein. We recently expanded the model to yeast 9. So, this is an ongoing process, continuously updated. We added more reactions. I'll not go into details on this. We also added a description of the protein secretory pathway in yeast that is also highly conserved to human cells and other mammalian cells. And we describe all the individual processes about how a protein is processed through the endoplasmic reticulum into also different compartments, how it's glycosylated and so on, and finally secreted out of the cell or left in the membrane. And we describe this again in the same modeling concept, and hereby we could actually capture also other important features of cell biology. But this concept I now explained to you for yeast, can we do the same for human cells? We can, and we did that. We developed a so-called, what we call human 1. Same concept, exactly the same way. Many more reactions, of course, many more enzymes as such. And of course, an important point here is that when we talk about human metabolism, that is, you could say, what is the capacity of any human cell. But in fact, that is different in many different cell types, right? So the metabolism in liver is different from that in muscle cells and that in fat cells and so on. But we can actually now begin to gather from this comprehensive model, fish out all the enzymes that are in liver and in muscle cells, and then begin to have a description of the metabolism in these different cell types. But let me just illustrate one example of use of the human model. So we were looking into whether metabolism of cancer cell is different from normal cells, and for this we used data where people had collected biopsy samples from cancer, 10 different types of cancers, and they have selected samples from both the cancer cells as well as from the normal cells, but from the same tissue. And then they looked at the differentiation, which genes are upregulated, which genes are downregulated. We could use that then to build models for the cancer cell and the normal cell in these 10 different type of cancers. When we looked at that, we actually were expecting to see some common trends in cancer transformation. But it turns out that cancer metabolism is actually in all cases more similar to the metabolism of the tissue of origin. And so there was not really any commonality as such in a cancer cell that could describe these phenomena. But one thing we did observe, though, and it was a little bit of a serendipity, that clear cell renal carcinoma, which is the most dominant form of kidney cancer, was really sticking out to be very, very distinct. And we saw that particularly when we looked at the metabolism, and here these are just expression of all the enzymes in clear cell renal carcinoma shown in yellow versus all the other 9 cancer types to the left. You can see a very, very distinct separation. And it turns out that there's several explanations for this, but it led us also to identification of a specific pathway, or set of pathways, I should rather say, involved in so-called glycosaminoglycans that are completely distinct, either up or down regulated in this cancer. And so this led us, of course, to think that we could, by measuring the associated metabolite with these pathways here, we could begin to find a biomarker. And in fact, we could. we found a really strong biomarker based on measurement of about 20 different metabolites. It's not in one metabolite alone, it's 20 metabolites. We use then machine learning to find a composite, and we call it a systems biomarker, composite of those 20 metabolites that can then be used as a score for cancer detection. This we have now pursued much further. It's been done, in more than 10,000 different samples from patients. We can detect cancers down to 1 centimeter of size, so that's very, very early detection. So that can, of course, be used for early cancer detection. But right now, we are particularly pursuing a strategy of monitoring for recurrence of people that have gone with after resection of their tumor in kidneys. And this is pursued now in clinical trial phase and hopefully will be available commercially in a year or so. So what about interaction with human cells and bacterial cells? Because we have a lot of microorganisms living in our gut, in our mouth, in many different places. How can we also begin to study this? And so we have a number of data that indicates that the human gut microbiome in particularly is changing with age, but also that it's very much associated with disease development. So we were looking into if we could use the same modeling framework for also studying these kind of interactions. So we did develop models for the most abundant bacteria in the gut, in the human gut. We combined this with an advanced model describing the human gastrointestinal system, and hereby we could begin to simulate what is the effect of changing in diet, for example. How does that change your gut microbiome composition, and how does that translate also to metabolites that you have in your blood? And this platform here now enables that we can begin to do many more complex experimentation and design of what probiotics should be used, for example, to repair dysbiosis in our gut microbiome. So let me end with a few phrases about lessons from AI. So I mentioned that we use information about the catalytic efficiency of these enzymes. Traditionally, we collected these from the databases and literature and so on. But with the availability of advanced AI models, we can actually now begin to predict these KCAT values. And so we developed a neural network for KCAT estimation based on just the primary sequence of amino acids. People were very skeptical about whether this would work. Just 2 years later, after we did this, AlphaFold came out and was published around the same time as our paper. But that actually helped tremendously in people believing that you can actually get these kind of predictions using AI. So now we can collect this. We've done this for any organism actually, and so we have collected in a database with more than 30,000 KCAT values for different enzymes that are available in different organisms across the different kingdoms. We also recently used AI to expand the coverage of yeast metabolome. I mentioned that all the reactions that we have in our models is based on what we tend to know. But if we look at all the different metabolites that have been measured in yeast, actually not many of them, it's only a very few of them that is covered by our yeast 9 model. So what we did here was that we looked into the promiscuity of enzymes for different metabolites, and then completely expanded, you could say, the model. And you can see here that what particularly leads to expansion is lipid metabolism, because there's so many different lipid species. But there's also many other different kind of chemicals that reactions that come up. And so hereby we can begin to map and identify byproducts of enzymes. And this is super important, particularly when we want to use these cell factories for production of different chemicals, because we don't want to have all these side products being produced. And so with this now, much more advanced models that were developed based on AI prediction of functioning of the individual enzymes, we can now have better description and design of cell factories. And finally, we also used AI for generating a new version of the human metabolic model. Here we use large language models to actually help in the curation of all the different reactions. So we have a lot of information, of course, from the literature, but we now are using AI to collecting and informing that and generating a much more comprehensive model. But we added also this, as I talked about before, develop models for different tissues and combine that with a circulation model so we can actually simulate how you go from nutritional input to functioning in the different tissues in the cell, and hereby also begin to understand what is resulting in dysbiosis or dysfunctioning of human metabolism globally. So with that, let me end. Thank all my students and funders over the years and you all for your attention. Thank you very much.